Hello and welcome, everyone. Great to see so many of you tuning in the morning for such a really interesting and important topic. My name is Kate Tarrant and I work for the Lower Blackwood Landcare Group. For those of you that don't know us, we are a not-for-profit and independent landcare organisation with an interest in sustainable agriculture and the broader environment and river health in the Lower Blackwood catchment. You can find out more about us and the work we do on our website, lowerblackwood.com.au. Before we kick off, I'd like to acknowledge the traditional owners of the land on which we all live and work. I acknowledge the Wadandi and Pilbara people of the Bibbulmun Nation and pay our respects to elders past, present and future. For those of you that tuned into previous webinars with us, you probably know the drill, but for newbies, I would just let you know how it's going to run today. You may have noticed that by now you can hear us but not speak yourself, and that's deliberate so we can keep things moving along at a good pace. You can definitely still ask questions though, so please don't be shy. Just type your question anytime into the chat box and I'll make sure it gets asked either during or at the end of the presentation. Also, if you run into any technical difficulties, please use the status button, which is the hand on the bottom right side of your screen, to let me know, and I'll see if I can fix it from our end. Don't worry though, if you have to leave early or do drop out, we will be recording the session and I will send it out to you later this week. Okay, to the reason we've all tuned in today, I'm delighted to have talking today internationally renowned and agroecologist, educator and systems thinker, Nicole Masters. Nicole has spent over 20 years working throughout Australasia and North America, coaching, consulting and advising producers on the approaches, tools and thinking required to build soil and ecosystem health, food quality and profitability. In 2019, Nicole added author to a list of accomplishments, publishing her first book, For the Love of Soil, which I have a copy of here. It's great for you, Nicole. It's a bestseller and has been described as a land manager's roadmap to healthy soil, revitalised food systems in challenging times. So if you haven't read it, you should. I guarantee that it'll become one of the most dog-eared books on your shelf. This is actually our second webinar with Nicole this year. And we know today's topic will be as fascinating as the last one. We've asked Nicole to talk to us today about the impact of chemical intervention on natural ecosystems and what tactics and strategies can be applied to efficiently and effectively control weeds whilst minimising ecosystem damage. So a very big welcome to you, Nicole, and I'll let you take it away.
Thank you so much for having me. Again, it's always a pleasure to speak to this group. I recognize a few names. Quite amazing to see you all. So thank you. Nice to see you, Fred. Yeah, and we will be talking about herbicide-resistant wild oats. So I know many you come from different different biomes. You come from cropping as well as working with animals or ecosystems, like natural ecosystems. So please feel free to add some notes. It's very helpful for me to know that this is something that you're interested in, or if you want me to kind of dive down different rabbit holes, I can always dive down rabbit holes. But I thought what we would start with was, what on earth is a weed anyway? So if we think about how weeds have been treated in more recent times, very much the paradigm of seeing weeds as a foe, so something that's going to reduce our farm productivity, something that's very costly to deal with, or something that's an ongoing, never-ending issue. And certainly what we're seeing is this development of resistance to our current treatments. So for many producers, just feeling like we're in a treadmill. So I want you to consider that in every cubic meter of soil, there are literally thousands of seeds, and they may be what we think of as weeds. They may be beneficials, they may be native plants, legumes, the whole lot. There is a huge seed bank sitting down there. And so what I'm interested in is, what is it that weeds are trying to tell us? And if we can figure out what their signals are, then how does that give us some access into some new actions? All right. So for me, a weed is just an indicator plant. It's not bad or good. It's really just a plant that we've got to start to ask the question of, what is it trying to say in this ecosystem? So we have plants that are going to quickly cover bare ground, so they'll germinate if there's bare ground, and they'll germinate if there's low organic matter. They might be responding to imbalances in minerals. It might be a microbial imbalance, or it might also be a safety valve for toxins, right? So I'm going to give you some ideas of how do we figure out which of these 5 things is a plant indicating, because then that can give us an access to what actions can I take newly. So one thing you can do is dig up a plant and look at its root system. What is it trying to correct? Often if we see large, deep tap rooting systems, they are what we call the dynamic accumulators. So these plants can be very high in different types of minerals that often the soil is very low in, or functionally low in. So think of chicory on the left. Fleabane on the right has this huge fibrous root system that really can make the most of low nutrient value. Things like purslane, so at that bottom right, you know, these plants that are scrambling. Anytime you see scrambling weeds, they're trying to cover that ground. They're trying to prevent soil losses, and they're often trying to prevent losses of carbon. So get interested in, you know, where do I see these kind of scrambling weeds? What might they be trying to tell me? Many of our broadleaf weeds may be indicating a functional imbalance between phosphorus and potassium. Generally, potassium is very high, phosphorus is very low, and that would be typical for your region. But asking this question, so looking deeper. So weeds as indicators. So many weed species are in fact indicating low available calcium and humus. Now you might be sitting on fairly high calcium, and I'm thinking of soil testing I've done along the West Coast, and I must say there's not a lot of high calcium that we come across, but what we're talking about is functional availability. And who makes calcium available is our active fungi in the soil. So taking a look at, do I have fungal activity, and then is calcium coming through? So many of our weeds, particularly primitive grasses, they're going to be indicating things like low calcium. Is it something that you're doing with your management? So compaction, overgrazing, or disturbance. Nut grasses, what are they trying to tell you? I was actually working on a project that was a 3-year composting project, and it was in an orchard, and they weren't particularly worried about the nut grass. It's not a problem in the orchards, but what was really interesting is that come year 3, although the seed, I guess they're bulbs, aren't they? Although the bulbs were still sitting in the soil, we saw no above ground activity in those nut sedges. That just through a process of building biological activity, lifting organic matter, we saw a shift away from those nut grasses. So one thing you want to try to start to do is reading your weeds. So take a tissue test of a weed or a least desirable plant versus your favored plant species. And then if it is a dynamic accumulator, we'll actually see it show up as very high in the nutrients that it's accumulating. So use this picture here. This is in the Tom Miner Basin here in yellow. It's just on the edge of Yellowstone National Park. And what's interesting is the bears are starting to come right down around the houses because they're chasing a plant called caraway. So caraway, you guys probably have this, I'm not sure. Anyway, it looks like a wild carrot. It's a Eurasian plant, and the caraway has been encroaching into these areas, and the bears love it because they dig it up, and then come fall, they actually are using that as part of their hibernation technique because it's high in starch, it's high in stored sugars. So we did some biological testing of these soils, and we did nutrient testing of these soils, and what we found was soils that were very high in boron and very fungal dominated didn't have any of the caraway, but where they were bacterially dominated and low in boron, that's where they saw the caraway. So they could actually take new actions by addressing this minerally and microbially so that the caraway wouldn't flourish, basically. So we did this not actually in the Blackwood Valley region, this particular test. So what we're looking at here is a nutrient test of ryegrass compared to capeweed. So the ryegrass is the first column, capeweed's the second, and what we're looking at is you want to compare and see where are they substantially different. So you go phosphorus, potassium, sulfur, carbon, all pretty similar until you get to calcium. So 3 times the amount of calcium in the capeweed, 10 times the amount of sodium in the capeweed, and then we looked at zinc, so twice the amount of zinc and 10 times the amount of boron. And then what was really interesting is double the nitrates. So I think most of you are aware that capeweed is a nitrate indicator. But what was really interesting is these leaf tissue tests correlated with the soil test. So the soil test showed low calcium, sodium, zinc, boron, and very high nitrates. And so the weeds are accumulating minerals which are low. So this dynamic accumulation means they're bringing those nutrients from lower in that soil profile and then returning that to the surface. And some weeds, like they've done studies on them here in the States, are actually returning those nutrients in a bioavailable form through their roots as well. Now, they could correct that, and you could allow them to correct that imbalance, and it might take them 100 years. Or you could ask the question of, what is it that this plant's trying to do, and how can I do it out of a job? So this, an application actually went onto this property with a bit of calcium, with a little bit of sodium, which probably seems really crazy, but some sea minerals were used in this case, some zinc and some boron, and then a carbon source to buffer and convert those nitrates. So asking the question of what is that weed trying to tell me, and just using a normal standard leaf tissue test, you want a complete analysis. So when you send this into the lab, ask them for things like sodium, trace elements, so you can get a picture. So nitrates, one of the things that bacteria do is they produce alkaline exudates. So anytime you have bare soil or you've had soil that's been dry for quite a long time, what we see is a buildup of these bacterial populations. And then there's some of these nitrifying bacteria. They release an enzyme to pull hydrogen off that ammonia, and they replace it with an oxygen, which creates nitrates. And so if there's no living green growing plant for a period of time, we see this buildup of nitrates. And then what happens is you could have bare soil, maybe you get a rainfall event or environmental conditions change. There's actually a signal that those seeds receive to say, hey, germinate, because most microbiology, most plant species, certainly human beings, we don't deal very well with nitrates, but there are specific weeds that are the nitrate weeds. So think pigweed, fat hen, nettles, thistles, nightshade, kochia, which I'd never really thought of as being in Western Australia, but apparently some of you do have it. Marshmallow, Russian thistle, foxtail barley. These are our nitrate weeds. So starting to look for, where do I see these growing, and maybe thinking, what do I need to change potentially in the system so that we don't see these species germinating and showing up in the system. All right, and then we have what we call the weeds of excess. So these excess nutrients, that could be nitrates, it could be very high potassium, there could be very high biological activity. So they're telling you there's some kind of imbalance in that nitrogen cycle. Potentially you've got low protozoa, so they are the predators that are helping to cycle those nutrients through consuming the bacteria, and low carbohydrates. So where we see low Brix, so low plant photosynthesis, those plants are not exuding a lot of nutrient or a lot of sugars at their root zone, and we see these weeds showing up. Some of that excess could be in terms of sodium. So you guys are probably familiar with some of these sodium weeds. They're able to draw that excess sodium up into their leaves. That plant on the left, I'm trying to think, like, is it a sea asparagus or something? Anyway, I've seen it growing on a few of my clients' properties now, and they're actually harvesting to sell to restaurants. So it's something to think about as a potential, and I have seen this in Western Australia. Now, interestingly, I've always thought of foxtail barley as being a nitrate weed. And then on a property in Reno, we were seeing it expanding down these valley floors, did the testing and found that actually it was mopping up excess sodium. So it has a mycorrhizae specifically adapted to address these high sodium issues, which is kind of cool. So it's actually sporulating mycorrhizae out that root zone. So if we do have an excess, potentially do we need to balance those soil minerals? Often the solution is, feeding fungi, building carbon. The addition of humates can be used, or humic acid, to actually help to bind to some of those excesses. You can make a protozoa tea. So you do that by soaking lucerne for 3 days, allowing all those protozoa to breed up, and then actually applying these. And they will actually help to balance some of these nutrients, in particular the nitrates. If you are applying compost, please, like manures, and you're composting it, make sure you're composting it. There's a lot of issues with people applying raw manures. I don't know if that's the case where you guys are. And then, yeah, using animals as a tool. I mean, your number one tool at the end of the day, if you have livestock, is using animals appropriately. So making sure you're moving stock more often and allowing for longer recoveries as long as possible. So I just want to show this case study. This is of mesquite. This was with Betsy Ross from Sustainable Growth Solutions in Texas. Unfortunately, Betsy just passed away. Incredible resource of knowledge. She was creating a database of what different plant species were indicating, what are they accumulating, and what she made up was a foliar application, and I'm actually going to give you that recipe. And she sprayed this foliar nutrient onto the whole foliarly on the leaves and on the bark and on everything of these mesquite trees. And what happened was this Cambrian, basically what you're seeing is the bark just expands and cracks. And these mesquite trees everywhere we went on her property were dead. So that by applying foliar nutrition, it actually did the mesquite out of a job, and it just died, which is a heck of a lot cheaper than some of the harsh chemicals that they're using on mesquite down in Texas. The same kind of approach could be used for your acacia, for some kind of brush. This is the recipe. Well, this is what's in the recipe. So 2.5 kilos of manganese sulfate. It's got 450 grams of Solubor, zinc sulfate, magnesium sulfate, and then that's mixed with a fulvic acid, and that's enough to do 25 acres. So very, very, very small amounts are actually signaling to these trees that you're not welcome here, which is fascinating. So this is the actual how to make this chelation. So it's a brewing process. It's very, it's like a slow, it's like rolling bread dough. It's this very slow kind of boil, just gently turning over. So I'll leave that recipe with you because I'm going to give these slides to Kate. And then if this is something you're interested in doing, we've seen some really great results with this on other crops, and it's very cheap to do. Right, so thinking about what are some of our biological indicators in the system. So if we have some kind of disturbance event, a disturbance event is going to drive different types of biological responses. Those disturbance events could be, invasive tillage, it could be fire, it could be pugging from livestock or flooding. My grandfather used to fly a DC-3, and he would top dress in that, if you can imagine. I wish I'd seen it, but top dressing superphosphate, and what they used to put in with the superphosphate was DDT. And so seeing the implications of DDT or DDE, which is the breakdown component, there's properties still experiencing that around the world. So that disturbance timeline, if you imagine, so this is not a plant succession in terms of evolution. This is a plant succession in terms of high disturbance to low disturbance. So if you can imagine a disturbance event that would take soil back to bare rock, and that might be a volcano or a really, really, really hot fire, a massive erosion, things like that. What you see is in those super high disturbed environments, they're very bacterially dominated. As these soils start to develop with plant cover, different, dust is collecting, microbials are reacting, and we're starting to really build soil, is that those soils move from super bacterial dominated to slightly bacterial dominated, which would be things like your annual grasses and your taprooted forbs, into a 1-to-1 ratio for your perennial grasses to 1-to-4. And that again depends on the biological test that you're using. So this is the initial work by Soil Food Web. So just check with your lab about where this ratio should be. But if you're wanting to grow orchards, especially deciduous orchards, that you might be looking at 1-to-10, 1-to-20 fungal domination, and then into more forested ecosystems. So consider what is the biological component that this particular crop needs and who's actually growing. And what you find is most operations are generally a mosaic. So there'll be undisturbed areas and overdisturbed areas. So you might have areas that are undergrazed, areas that are overgrazed, or areas that are undisturbed because they're under a fence line, something like that. And what we see is huge domination of bacteria. So most of the microbial testing I've done in Western Australia is highly bacterially dominated. And then that leads the germination signal for these specific weed species. So in this example here from the Soil Food Web in New Zealand, we've got the 1-to-1 ratio for grass at the top left. They come in, they cultivated and sowed kale, and we're at that 0.75, perfect ratio for kale. They then came in after the kale, cultivated again, and reduced that biological community to 0.5, and then what you see are these early successional weeds. So this could be, in this case, it might be, I think it's cat's ear, that could be dandelion, but any early successional weeds, right? So the more bacterial we push, then the more challenging it is to address these weeds. They get harder and harder to kill. And so I saw this, this is probably one of the best examples of the difference that we can make with changing the biological community. So I saw this project last year in Cotopaxi in Colorado. It's Steve and Nancy Oswald's property. This photo here is of some of their low-lying meadows. These meadows tested fungal dominant, huge amount of diversity in these meadows. Just absolutely beautiful to see, lots of native grasses and all the rest, and animals do very well in here. They had some fields that they call the cheatgrass fields, and all that these fields would grow was cheatgrass and purslane. So cheatgrass is a very primitive grass. It's kind of palatable to livestock early in the season, but it's not good animal feed later on, and it's a fire risk. So it's an annual grass, has a huge amount of seed production. For those of you that you're probably not familiar with it, but so what they did is they did a trial applying 25 pounds an acre of a Johnson Sioux, which is a high fungal compost, and they applied that as an extract. So what you're seeing here are the test plots. So over 1 meter squared plots, they applied the extract, and what you're seeing around those plots is just the purslane and the cheatgrass. They planted the cover crop across the whole field, and the only place that these germinated was where they received this biological treatment. Now, what was interesting is they did the same thing in the meadows, and they got zero response. They didn't see any change where they had applied this in a fungal meadow, but in the highly bacterial soils, you can literally see where that fungal extract went. So something like this is very, very cost effective, and I can see being used anywhere that you are able to actually put applications down. But yeah, it's a really cool project, and I think they're doing even more this year. And then this is an image of what it looked like in that soil where the sorghum Sudan was growing. And you can see that that soil is darkening in front of your eyes. You can see those, what we call the Rastafarian roots, right? So soil sticking to that root surface. That single treatment has now kickstarted that soil. So yeah, you should be able to start stepping back a little bit with that soil starting to function. It's really cool. So if we leave soils undisturbed, and so disturbance could be, like I said, it could be fire, it could be a severe drought. But if we leave soils undisturbed, then we start to see that biological succession start to advance. And so we might start to see things like your blackberry. I have never done the testing on arum lily. I wonder if it is a, not sure on arum lily, actually. I need to do the research. You could see things like bracken, low growing shrubs, spiky things, right? So this is the soils become more fungal, but I think of these soils actually as sleepy, right? So they're starting to have less activity, and then that sends the germination signal for these sort of shrubby vegetation. This is an example of taking care of some of that shrubby vegetation, and in this case, it's sagebrush, which is very common out here in the West. Yeah, so right now I'm actually in Nevada, but normally I'm based in Montana. Haven't been to Australia since March 2020, so hopefully things get easier to travel, because I miss being in Australia. But what the rancher here is doing is actually spreading hay bales on top of the sagebrush, and the cattle get in there and beat it up. That sagebrush does not like disturbance, does not like being stood on, and these sagebrush bushes are dying, and then that's returning back to pasture. Just cool. So cool. All right, so the other weeds of excess, we can actually have an excess in minerals, but we can also have a toxin release. So I'm interested in Bathurst burr. I think it would be an interesting one to take a look at. You know, when we do have low organic matter in soils, then you have no buffer against agri-chemicals. So things like drenches, porons, there could be historic heavy metals, cadmium, things like that, start to become available when that soil organic matter becomes degraded. And so I think Bathurst burr potentially is doing something to release those toxins, and part of what it's doing is actually telling you to go away. Same with milk thistle. So thinking of milk thistle like it is in the human body in terms of it's a it's a plant that we use as a liver detox. So seeing properties where this shows up is where there potentially is some kind of toxin, and that the soil is basically trying to regenerate and rehabilitate. And the first time I really saw this was in New Zealand after the 2010 earthquakes. And it was interesting. You know how you sit on a plane and you don't really talk to people? It's always a bit weird. It's always interesting to me who I do end up talking to. And this one time was a nuclear physicist. And we were talking about the earthquakes. And one of the things that happens is deep down in the plates, when those plates grind and you get a large earthquake, there's uranium down there, and what's released is radon gas. And what we were seeing in New Zealand popping up along these fissures was milk thistle. So the milk thistle was effectively mopping up that radon gas. And this photo here is taken in California, and this milk thistle is 8 feet high. And when we went to look on radon maps, this is a hot spot for radon. So those plants were literally trying to get that out of the soil environment. So it's a really interesting, I think it's really interesting just to ask the question of what is this plant trying to do? So things that we can do with those release valve weeds, you want to test your pastures with a refractometer. If your refractometer measures a 3 or below and it's a sharp line, that's telling you that you do have nitrates in the system. Avoid grazing if you're seeing a lot of these release valve weeds. Test for potential contamination, or ask the question of how was this land managed in the past? You know, is this an area that it is a sacrifice area, or there was a lot of ag chemical used here? What's happening with soil carbon? And then using inputs like humates or biochar or milk thistle, we see can have a really interesting effect in helping to remediate. Fungi really are your number one remediators. I mean, they use oyster mushrooms right now at the Fukushima radiation site. So they are actually putting oyster mushrooms around that zone to mop up some of that radiation. They can do a great job with cleaning up a lot of heavy metals. You could also plant things like sunflowers or willows or plant species that are known to bioremediate, and build your soil carbon. That's going to be one of your biggest actions. So question here on wild radish. So wild radish is included in this non-mycorrhizal category. So take a look if you see a lot of non-mycorrhizal species, so pigweeds, lamb's quarters, mustard, shepherd's purse, that's a little love heart one. Love it. Russian thistles, lupins, kochia, sedges, rushes. If you're seeing a lot of these species, then be asking the question of what am I doing to disrupt fungi in this system? For pigweed and lamb's quarters, it might be actually you're putting too much nutrient out, or there's a concentration of an excess of phosphorus and nitrogen. for some of these other species that potentially, yeah, something's happening to disrupt that fungi. Is it a disturbance? Have you been out tilling? Have you undermined your mycorrhizae? So doing mycorrhizal testing on your particular crop. So your wild radish really is a very bacterial dominated soil, and very non-mycorrhizal, right? So asking that question. Calcium is also associated with your wild radish. So looking at how do we get calcium functional. Right. I don't know what happened with this one. So yeah, addressing weeds at their root. So literally going to the root to ask that question. So as a case study, Alder Spring Ranch, which is in May, Idaho. I've actually just been there for the last month. It's fabulous. They're 46,000 acres. They're one of the first regenerative organic certified. They practice what they call in-herding. So it's a type of shepherding where there's 4 to 7 interns who are moving and living with those cattle every day. They bring the cattle in at night into night pens, and they have GPS on the riders so that they ensure there's no repeat bite of these plants for 1 to 6 years. So they're going out towards water and then coming back to the night pen at night. And it's extraordinary to watch. And just had to put a gratuitous picture of myself and my dog and my horse. But you see the country that we're riding through. It's absolutely nuts. It's a volcanic tuff. It's huge amounts of shale. There's a lot of bare ground. But what was fascinating is over 2 days, I counted 127 different plant species. And out of that, none of them were non-native. And in fact, to find non-native species, this is one of the most pristine prairies I've ever been on, by far. The only place that we saw non-native species was where there had been fire, and they brought in bulldozers, and everywhere that the tracks were was where you would see these invasive plant species. But what they're creating is an ecosystem of natives. So we see incredible native diversity up here, all sorts of flowering plants. Some of these are edible. Cattle think they're all pretty darn edible. Now, what's interesting in this process of sort of just moving across a landscape, they've lifted their organic matter from 2% to 4.5% in 6 years, which is absolutely extraordinary. There's been a 300% increase in ground cover and in plant diversity. So they set up some transects just to look at that ground cover, and just extraordinary what is germinating in this ecosystem through the basic, just using animals as your tool to enliven and awaken up those soils. You've got manure and urine, but you're also supporting the health and the grass populations. If you see in the foreground, that grass has been grazed. Animals have moved across that, and now potentially they're not going to come back. So this idea of making sure that animals are not just sitting around and taking plants right down and damaging what is a fairly fragile ecosystem. So another example of addressing a weed concern in this situation, and I talk about them a lot, they are my American family. I've been working with them since 2013, is they always had these fields that they called the farm ground fields, that were incredibly degraded. So all that was growing in here is a yellow alyssum and a little bit of some fescue, but very low species diversity. And it had been like that for 30 years. So it had historically been farm ground. 30 years ago, they had come in and cultivated that up, and then pretty much nothing had happened. It's basically blow dirt, like just super structureless, falling apart. Awful. So what we did, and this is something I recommend that you do, is look at improving your good ground first. So they invested in a soil health program on their irrigated ground, and then use that additional hay to pay for the investment that they put into this farm ground, so that they're not out of pocket, right? And so the program that we put on was based on their soil and leaf tissue tests, and what we put on was, I do go a little bit crazy sometimes and mix stuff up, and maybe this would be a nightmare for somebody, but it was worth it for me, maybe not for them. Oh, maybe this video is not going to play. Pointer, point. I have a private pointer. Well, that's fancy. No, the video is not going to play. Sometimes with different types of technology, it can be tricky to get.
Oh, yeah, the video won't play as an embed. So if you wanted to share your screen, we could see it if you played it separately.
Okay. Maybe look. I think I could do it. MP4. No, it'll take me too long to find it. What I wanted to show you is this particular setup. So Roger and Betsy Ingerland created their own slurry sprayer, which is what he's trying behind that tractor. That tank will do, it's 4,000 liters. Oh. It's the thing with gallons, liters. I always say to the Americans, they need to learn metric. Anyway, that tank is enough to cover 15 acres of treatment, and it can take like a considerable amount of solid material, and you can also put seed into it, which is kind of cool. So they did a this mix on the dry land. So 10 kilos of Epsom salts, 10 kilos of a mined rock salt. That's very popular here. They did 2 kilos of Solubor, 20 liters of a fish hydrolysate. The fish products here in America aren't as good as Australian products, so you could probably use less. 5 cups of sugar, 5 kilos of vermicast. They're making their own vermicast. 5 kilos of manganese sulfate, 2 kilos of kelp, 1 tablespoon of yeast, and then we put in some some vitamins, including some milk thistle. So the milk thistle and the yeast themselves are known to be bioremediators of chemicals. If you have chemicals in the system, yeast actually has the ability to break down atrazine, tiny amounts. So research has shown that you only need, like, if you've got those little sachets that you get to make bread, you only need 1 sachet per acre to create a response. They did some work in sugar beet and found that they could lift sugar beet sugar production by 23% just through the addition of this tiny amount of yeast, which is kind of cool. All right, so that slurry sprayer has a nozzle size that's 1/5. Well, it's I can use metric. This is a joyful thing. 5 millimeters in size, so you can put seed out in this. So clover seeds, lucerne, a lot of grasses that are smaller will fit through the spray nozzle, and you can spray it out, which is really cool. This was the plant tissue testing after the application. So they did sow a hairy vetch and a rye into that field with this treatment. And so what I've highlighted is where it's significantly different. So the nitrogen came up, the phosphorus came up, the calcium came up, the magnesium doubled, and these are now all in range. The boron came up. And what was really interesting is this region has huge problems with excess of molybdenum, and it causes animal health issues because excess of molybdenum will lock up your selenium and your copper. And this treatment dropped that 7-fold. We have a vet who works with this particular operation, and she said this is a game changer for Montana if they're able to drop that molybdenum because it causes such issues. You can't give a bolus to livestock. You actually have to inject them to be able to get nutrition into those animals. You know what? I am going to have to show you this because this won't work by itself. And so this is us driving across this field after the application. Now, admittedly, this was the best rainfall season that they had there for a long time. Like 2018 was an absolutely fantastic year. But it's not just that moisture. So in 30 years, this field has really grown nothing. And, oh good, it hasn't got any sound.
And
I can just share screen, can't I?
Yeah, Michelle, Nicole, down that's on the menu side of the screen, on the I think one fourth icon down is a screen sharing button.
Yay! Is it playing? Okay, I'll play it. So yeah, we're driving across a field that's now got alfalfa in it. Oh, I'm sorry, lucerne. You can see that rye. A whole lot of native grasses came up. There's sweet clover that responded to the moisture, but just this incredible diversity in a field that has been absolutely miserable for as long as memory. It was always an area that they didn't want to do anything on. They felt like it wasn't worth investing in. How do I go back? I'll present. Aha. So fancy, Kate, so fancy. And what's interesting for us is to go back and dig holes and see the difference in the soil. And I really need to update my database because the changes that have happened in the soil from a blow dirt that had no structure has now got structure. It's now infiltrating water. And they don't need to come back and do another treatment. This is a one-off treatment that's addressed that weed pressure, that's changed that biological signal to what is now germinating is that latent seed bank. And as long as they continue to manage this well, and these guys are really good managers, then the plants are now setting that scene for health. You know, once that plant is growing and it's got a healthy root system and it's able to access nutrition through that biological bridge, you can step back. So they are not going to have to intervene again in these fields, that they're now going to keep on rocking on. Right, so being able to figure out for yourself what is what's going to work well in my system, do these little test plots. So I learned this from Dr. Arden Anderson, and we use this a lot. So set up little 1 meter by 1 meter squares, and you can just do that with electric fence posts. Get a little bit of water, like 100 mils of water, and then put in the equivalent of what you might be applying. So that might be like 0.5 of a gram of Epsom salts, right? Or it might be a couple of drops of, or it might be 4 mils of fish, for instance. You're going to spray that with your water and then come back after 40 minutes and look at what has given the highest response in terms of Brix. And you want to see at least 2 Brix higher. Now we did this at the Indralins, and the Brix actually dropped by 3. And what we'd put in was 30 pounds of vermicast per acre. And with that, so that's 30 kilos a hectare. So what we realized is we'd put too much on, but we tested these plots a week later and their Brix had gone from 10 to 20. So you want to make sure you have a control so it's not responding to water, and take a look at, is this product actually going to enhance plant photosynthesis and set me up for success, or is it going to drop it down? And at the meantime, you're going to be testing your weeds. So you might have kochia, or you might have or you might have capeweed. You can measure your capeweed, and what you'll see is if the Brix in the grasses go up, the Brix in the weeds will go down. And so you want to see that because potentially what's happening on most operations is your weed is photosynthesizing higher than the crop that you want to grow. So who's winning the game of life? Not the crop you're wanting to grow. You are literally farming for weeds. So you need to assess what is it that's going to help jumpstart this system, and what's going to flip this switch. Right. So then thinking about buffering our chemicals, or just looking at how to reduce some of these herbicides. So you can reduce herbicide and pesticide use by 10 to 30% by decreasing the pH. And you can do that with a citric or a fulvic acid and get the same efficacy. Now you'll find on most chemical supply websites, there is a list of, what pH will this perform best in. So just check. Also check the hardness of your water to make sure that your water's not causing an issue, which is why you're needing to use more herbicide than what's on the label. lift that functional calcium, all right, and lift biological fungal activity so that you can address not only your grass weed pressures, but a lot of weed pressures. So it means if you're in a cropping situation, just maybe you're going to put out small amounts of calcium into your program. Lifting available phosphorus, who does that? That's the role of mycorrhizal fungi and fungi again in the system to help lift phosphorus. If you are going to spray, maybe spot spraying instead of boom spraying. And then also you can buffer your chemicals with some kind of carbon input. I really like fulvic and humic acids. I use them a lot. There's fulvic and humic acids in compost extracts and vermicast extracts. What they do is they lift cell wall permeability. So what that means is they increase the efficacy of foliars or herbicides into the actual plant cell walls. So it means that if you add in a fulvic or a humic, you can drop your rate, in particular dropping glyphosate by 30%, and then adding in a fulvic acid. So 1 part fulvic to 4 parts glyphosate. You can reduce your herbicide use, but what you're also doing is you're stimulating the microbiology that can actually work to break down some of the chemicals that are sitting in your soil system. So you're stimulating that microbiology because many of them, if they're able to be stimulated and they're present in the soil, are able to break down and remediate some of these chemicals, right? So if we consider herbicide resistance is just going through the roof, and obviously this means that we need to now genetically engineer plants for dicamba and 2,4-D. That's obviously the solution, or a bigger hammer. All of these bigger hammers aren't working, and what's happening is there's an epigenetic response that's happening inside plants, is that some plants can overexpress the genes that relate to the particular mode of action that herbicide's working on. So for glyphosate, for instance, so glyphosate resistant weeds overexpress the shikimate pathway, which is the pathway that glyphosate works on. Joe, did you have a question? If you have a question, I'm happy to take any questions. So we see this overexpression of this pathway, which means then that when the glyphosate's acting on the shikimate pathway, this plant is still able to overcome that and survive despite this effect, right? So we now, if we look at what's happening here in the US, 29% of cropped land has herbicide-resistant weeds covering 24.5 million acres, and these are our wild oak, the green foxtail, and broadleaf weeds. Getting a bigger hammer is not working, and there's some interesting stuff in terms of epigenetic alterations, so the DNA methylation that is how plants, if they're exposed to this, they don't die, they then pass that gene expression on to future generations. But what's also interesting is aromatic amino acids can interrupt this overexpression of the gene. And aromatic amino acids, they could be included in things like fish. There could be some of these in our worm extracts, which might explain what Di and Ian Haggerty is seeing. So you guys are probably all familiar with them. 45,000 acres up in Waikato, Western Australia, sheep and cropping, 8-inch average rainfall. What they're doing is, after grazing, if it's possible to get sheep across all the land, they are drilling their seed with a worm extract or a worm wash. They're applying 5 liters per hectare at seeding. In ground that they've taken on that has huge herbicide-resistant weed pressure, they're doing 120 liters a hectare of a compost extract in the growing season. So when they first took on some of these blocks that are full of probably some of the most persistent, resistant ag chemicals, what they saw in their first year after the crop was harvested, that they saw a lot of primitive grasses like your button grass, your kerosene grasses, what I think of as crack grass, windmill grass, seeing those kind of species germinate. And then in year 2, they saw a shift to Paspalidium grasses and native C4 species through changing that biological signal. Now, what's super cool is they sent away samples of the soil that had received these treatments for 2 years and samples of soil that had not received any of the treatment. And what they found, so Prospect is the name of Di and Ian's farm, is that seed that had been exposed to this vermi wash, the worm extract and the compost extract, was killed at half the rate of glyphosate and half the rate of trifurulin, whereas the seed that was sitting in the soil that hadn't been treated at all was still 50% is still surviving, or even 80% was still surviving at twice the recommended rate of herbicide, right? So in part, this could be explained by microbiology. In part, this could be explained by the switching off of this gene expression. And as yet, and maybe they've had some more updates, but we're not seeing interest in trying to discover what is the phenomenon, what's happening here on this property, because we see this again and again and again. So I was working with Kokako Vineyards in New Zealand in 2006. When I started working with them, they were doing 6 herbicides a year. It was old potato ground. Those weeds were still growing all up into the canopy. And within year 2, they were down to 1, if not 0 herbicide applications, just through stimulating microbiology, putting out a really nice biological product, actually, that had a little bit of trace elements, had some yeasts, had some of this microbiology that are known to bioremediate soils. And herbicide problem went away. So there are some other things that you could look at doing. You can harvest a weed extract. So take 5 to 25 kilos of raw weed per hectare. That's what you're aiming for. That's what the research literature kind of says. So you're going to take fresh green plant material. You're going to fill it in a drum, like a 200-liter drum, and then put a really heavy, like concrete weight on top of it to squish it. You're not adding any water. This is not one of those stinking comfrey ferments. And what's going to happen is it's going to squeeze down and start turning into something that kind of looks like slippery old Marmite. What it contains is the concentrated minerals. It's got pathogens in it. It's got allelopathic compounds, and it's also got enzymes. And you can use this to spot spray. We've seen this be incredibly effective as a herbicide. And in some cases where it hasn't been effective, we've seen effects like this. So this was a bindweed extract that Patty Armbruster up on the High Line in Montana made, and she just dumped it as a concentrate. And what's super interesting is you can see the difference where it was dumped and where it wasn't dumped, but this greening effect, because what these extracts is really, really super high in is plant growth enzymes. So we can see the concentration of plant growth enzymes, and we can see this allelopathic effect. So depending on what the type of weed was that you were growing. So I think that's given us just a little bit of an insight into reading what your weeds are trying to tell you. Now you can take your eye off the weed and focus on what we want to grow. And if you want to get rid of weeds overnight, we could just stop calling them weeds and then they've gone away. But Kate, I'd love to hear some questions. And if people are interested in expanding on this a bit more, there's a whole chapter on this in my book.
Yeah, fantastic again, Nicole. Really, it's always such a dilemma controlling weeds, especially the more you become aware about the role that weeds have in nature, should we get them to do their job? And the potential harm that we can obviously do to ecosystems by using chemicals. Yeah, I think your presentation has really helped with some of those decisions. But while people are thinking about their questions, I know there's a few there, but I'm going to get them first, being the host.
Can I press close so we can just see each other? Does that work? Yeah.
Oh, we don't come up big. Not like Zoom. We're still small.
Not like Zoom. We're just little boxes. That's fine. I'll make you so I can see you.
Just it's probably more of an overarching question, but talking about sort of bacterial dominated and fungal dominated soils, and sort of, you kind of got to be careful not to get the impression that bacteria per se are bad, because they're not, are they? It's just more that fungi aren't present because of perhaps management practices or other reasons. And so getting fungi into those bacterial dominant soils will go a long way, as you say, to helping eliminate the environment for weeds to thrive in. You don't mention you didn't mention really in presentation sort of the inclusion of cover crops or adding more plant diversity to help counter.
No, and we can certainly talk about that now. Yeah, and I think, so if you go back, just what your first point was, I think is really important. You know, bacteria are not bad per se. What we see is a loss of diversity and biodiversity in those bacterial populations, and we can see a lot of bacterial pathogens when we lose diversity. And then that feeds in really nicely to things like cover crops, is that those cover crops can drive that diversity of microbiology. So we know legumes feed a wide range of organisms, in particular clovers and other types of legumes. We can use C4 grasses, for instance, or C4 species to really stimulate mycorrhizal sporulation, so getting more mycorrhizae in the system by using cover crops. Yeah, I mean, the Ingeland example that I used, they put in a rye and vetch mix to help to stimulate this pump and to get the microbiology active. And really, this is what's missing in all of agriculture, is how do we get more diversity into the system, because a diverse ecosystem is going to be less susceptible to weeds, pests, and diseases. But yeah, I think interrupting cycles, you guys are in a higher rainfall environment than a lot of places I work in, where cover crops can be really risky in terms of thinking about moisture levels for germination.
Well, we have we're doing quite a bit of work in that area, but of course the challenge here is that 99% of pastures are annual systems, ryegrass and clover, and of course with the Mediterranean summer we don't have much growing over that period. So it's trying to put more perenniality into the system so that we get living roots over the summer, even if they're dormant. So that's a process for us to go through. But and my second question was just related to the weed extract tea you talked about. So will that work on nitrate accumulators then as well?
Yeah.
It will.
Yep. So give it a go, and just try it on these different plant species. I mean, I think anything that is an accumulator, we had some great results with it on thistles, because it bred up a whole lot of thistle rust. And so basically what we were brewing was thistle rust. It was pretty cool.
Great. All right. I'm going to go to the question back now. We talked about what herbicide resistance in wild oats, Fred, so hopefully that's giving you an answer. Sonia has asked about double Gs. You didn't mention that specifically. Are you aware of double Gs? Do you know what they are?
Double Gs, tell me more.
They're a really nasty, thorny, they have a huge thorn to them, so you can't, they can puncture tires, basically.
Is that the one that's got like the triple seed head with the spikes on it?
Yeah, that's it.
Yeah, that's quite horrible. You guys can keep that one. But yeah, I mean, it's interesting to me. I've said interesting a lot. I find this whole thing fascinating. But those kind of plant species are almost telling us to get off the land, and get off. Why? Because we're degrading organic matter, or we have really low carbon in these soils, that they are in a lot of ways trying to protect that environment. And I saw it really markedly when I was at Martin Royd's place in Jillamatong. He was getting us to feel his grass, which is really lovely and soft. It's like, oh, beautiful grass. And then he made us jump the fence to the neighbors, and it was only like 2 meters away. And the minute I put my hand down, and I put my hand down hard because I've been hitting his grass. It was covered in spikes, and it was literally like, get off, get off, get off. But I think a lot of these, spiky plants, they can be really high in silica. That silica is becoming available, or those plants are accumulating it in part because of the low carbon in these soils. So just thinking about, how do we just build soil health,
So back to more diversity, and maybe try spot spraying with your weed extract. Maybe that might work with double J, perhaps.
Yeah. I mean, I don't see why it wouldn't. It would be really fun. If your kids are really naughty, then that's going to be their school holiday project, is to go collect that stuff and make an extract.
Katie is asking how to best efficiently restore tracts of grazing pastures into natural bushland areas and habitat corridors.
Yeah, it's a good question, Katie. And I think if you look at like early historical accounts and ask the question of what was here, you guys had a lot of bush and scrub and forested areas. Would that be right?
Yep, most of it was already forested here. Yeah,
and so my father and I did a project in 2000, and what we did is we drenched trees, and we were planting trees, and after we planted them all, in came the blackberry and in came the gorse. So I don't know if you guys have gorse, but you certainly have blackberries. And we thought we'd wasted, like, we planted thousands of trees and thought, well, that was a total waste of time. But that blackberry nursed those plant species and the gorse nursed it. And because, it's probably been in pasture for maybe 50 years, the native seed bank was still there. And just by stepping away, you can see photographs of my dad's place now, and it's a standing forest. It was admittedly in a place called the Bay of Plenty, so things sort of happened quite fast in New Zealand. But it was a remarkable example of just allowing that biological community to shift. And what stops it shifting can be climate. So seeing this increasing drying phase. So I certainly would be doing what we call biopriming of seed. So putting vermicast and compost extracts on those seeds, or on the root ball if you are going to be trying to get forested tree areas back in, and just fencing it off. And just allowing that restful phase, and you'll be amazed at what starts to germinate, really.
And Katie, also give us a call at the Landcare office, because we do a lot of revegetation work, not just in riparian areas. So have a chat with us. We might be able to give you some advice and some help. Jason Daniel from Alberta. Hello, from Canada.
We've got quite a few Albertans on.
Yeah. Canadian thistle. I don't know that one. But
we call it Californian thistle.
Okay.
Cali's.
Are all thistles equal? Same treatment for all of them, is it?
Yeah. So if you had Canadian thistle, then Canadians need to get onto this. So the rest of the world calls them Californian thistles. So we should call them Californians. But it works very, very well. You just want to collect one species. So just collect either just Canadian, or just bull thistles, or just milk thistle, whatever you're seeing. But yeah, again, I would still be sending in a plant tissue test out of curiosity, like what is it trying to say? But yeah, any thistles.
Serrated tussock control, it's relentless. We're keeping greater than 1,500 kilograms of biomass in introduced pastures, and 800 to 1,500 kilograms on native Aussie pastures. Rotating stock, blackberries rapidly catching up, tussock levels of invasion, farmers in steep rocky country. Yeah,
that's the trick, really. It's so challenging to get animal impact. I haven't worked much with serrated tussock. I think it would be an interesting one to do this weed extract with. But if you're really seeing blackberries encroaching, then it's saying something about that rotating stock in terms of impact, that you're still not getting adequate impact in the system. Yeah. Yeah. I'd be taking a look, you know, and digging up that serrated tussock. And the places we've dug it up, it hasn't got a very well developed rooting system. But I don't know if that's the case everywhere. So dig it up, take a look, and yeah, do the leaf tissue testing on it. What's it trying to do? Yeah, and if it does have a very shallow rooting system, that's often indicating that overgrazing can help stimulate it, because it's not going to compete well with deeper rooting plant species. But it sounds like you are allowing adequate recovery.
Interesting. We've had Dr. Judy Earle over recently. She's a holistic grazing educator, and some of our people over here have got African love grass, which I guess is a kind of a tussocky grass, similar sort of thing. But anyway, she was saying, of course, they thrive in poor soils, and the best thing to do is disturb them. Lots of disturbance. So I guess if it's possible on your steep, I don't know how easy it is on your steep rocky country, but to try and get a little bit more mob intensity, so that you're really having major disturbance, and that probably help with the blackberry as well, I imagine.
Yeah, that's right. And that's why I think it's important for us all to be traveling and just see what some people are dealing with. I mean, we're working out here on some extraordinary rocky, intense landscapes, and how people are still able to manage impact. We're also seeing the use of collars over here, so the electric collars to control some of that really, really, really rocky stuff, and seeing some great early responses anyway.
Yeah, in actual fact, just that reminds me of something for our West Australian Southwest audience. The Talk and Soil conference that's coming up in York. There's a chap who's going to be speaking about virtual fencing and using that for mob grazing. So you might want to go along and listen to that. That's of interest to you.
That would be fun.
You've already answered the question about wild radish. I've answered the question about the slides. Okay, Fred asking, compost extract like you used in Cotopaxi, how many pounds, I should say kilograms, Fred, of compost and how many litres of water? So why do you do that, pounds and litres? And how many litres of that per acre?
Canadians live in the in-between world of having to deal with America. They talk pounds and litres, and they also talk kilos per acre. It's very confusing. So they were using 35 pounds of compost an acre, and it's going to depend on how much water, depending on your spray equipment. So yeah, if it's going in, if you're able to put that fully into solution and it's sieved out well, then you can put a little less water on. But yeah, this could be 400 liters to 1,000 liters a hectare, so 300 liters an acre ish. 400, yeah. Yeah, so it depends on your spray equipment. Fred, you can put it on pretty concentrate.
And then the next question, how to control blackberries in hard to reach places? Goats?
Goat deficiency. Or fire, but we shouldn't say that very loudly. We just had a big fire out here. We just had 24,000 acres burn. So,
yeah.
But that's going to take care of some stuff. That nutritional spray, the recipe that I've given you, I think would be really effective. And so you still are going to need to be able to spray or get in there somehow. My dad hired this incredible mower mulcher that was driven by a drone to go down incredible steep sides, and it just mulched like he had woolly nightshade that was like, do you get woolly nightshade?
I don't know. I don't know what it looks like.
You're lucky if you don't. Anyway, it was probably like 4 meters. Anyway, tall, can't think anymore in metric or not. Anyway, yeah, and they just drove this thing down these really steep sides, and it obliterated all of the blackberry and the woolly nightshade. It was incredible to watch, and it was all like remote control. But yeah, it really needs a disturbance Patricia, and that's the trick, is how do we get a disturbance in an area that hasn't been disturbed, and then ensure that you keep up that disturbance. Otherwise, it's going to revert back to blackberries or bracken or whatever it is that starts growing in your area. So yeah, you got to figure out, how am I going to disturb it, because perennial species require some kind of hard disturbance. Yeah, and some of those spray nozzles, we can get them out pretty wide if it's off the edge or down by a stream. But it's sure a lot better to be spraying these nutritional sprays than some of the chemical that they're putting now in riparian zones. I really wish people would stop doing that.
Well, that's why we've got this webinar, Nicole, so you can tell us about
just cut it out. Yeah.
Interesting. I saw a documentary recently, and I can't remember what it was from, but using drones for spraying in hard to reach places. So
Yeah,
that's a potential in the future as well.
yes, and people are doing that already. I've got an island client that was doing that. It was amazing, just to fly over and spot spray.
Yeah.
Yeah.
Shorty Fensky has asked, can Nicole explain what happens when too much fulvic acid is used with a herbicide?
Yeah, but I mean, and you'd have to put quite a bit on, but what can happen is it can totally bind up that herbicide, and then you're not going to see efficacy. And also, because the microbial population in the soil is going to feed first, then we can see a shock to the microbial system as well. Potentially that fulvic acid, any carbon needs to balance itself with the nitrogen, so we'll see nitrogen draw down from plants. So yeah, don't go too much, and then this is the tendency, I think, for people to think we need to put a lot on. Everything we're talking about is almost parts per trillion. Like if you look at the nutritional spray that I'm giving you, it's tiny, it's grams per hectare. Like it's just phenomenal how little we need in order to create a response. So don't go nuts with the fulvic acid, and don't go nuts with compost extracts or any of that.
And I don't know if you've actually answered this, but when using fulvic acid, does the maximum residue limit for any particular herbicide change?
I don't know the answer to that. We are, however, seeing crops that are using this technique turning up with nil residue. And Shorty, you might be seeing this yourself, but up on the Highline, producers that have been using this technique are seeing that they have no residue for herbicides on their crops, and organic crops are being rejected because organic crops are having herbicide come down in the rainfall and coming in the dust. But those that are using fulvic acids and the worm extracts are seeing that stimulation of microbiology actually is meaning that their crops are cleaner than organic.
Yeah, and actually, Grant Sims, you know him, don't you?
Oh,
yeah. He's been doing quite a lot of work in that area, and he was saying quite recently that they've just done some tests to show that, because I think he adds fulvic with his glyphosate to terminate his crops, and he said there's been no residue whatsoever of glyphosate when they've done tests after 2 months or something. It's all gone.
Yes. No, we've been, just blown away really with the nil residue and the digestibility and other quality measures despite using herbicides. I often share a story of an orchard I was working with in 2006 that had a scientific study done on it, and they applied 2,4-D to their soil, and when they measured it, there was no 2,4-D in it. They repeated the spray, and again, no 2,4-D. And what they'd found was that the microbiome, if you have a healthy microbial population and all of that diversity in there, the microbes can actually deal with a lot of this stuff. Even some studies coming out about atrazine and the role of yeast, different types of microbiology that will actually bioremediate. But what we have are ecosystems that are so degraded, they don't have the ability to remediate. So yeah, the fulvic acid's going to help, or worm extracts is going to help in that.
Okay, and Nicole has asked if you have any suggestions on how to manage onion weed, or what onion weeds are telling me about our soil.
Yeah, onion weed and I think nutgrass, probably fairly similar indicators. biological activity, calcium, building organic matter. But yeah, dig holes, take a look at your soil test. What are you low in? What are you really high in? And send off a little leaf test for those onion weeds and take a look. But yeah, like I said, we've seen some pretty dramatic responses with a very high fungal compost applications, and seeing them just not sprouting bulbs there, but not sprouting.
And Jason's asked about making the tea. You put the plants in the bucket without water, just a bit to weigh it down. How long does it take?
It'll be bigger than a brick, like a big block of concrete. You're going to harvest those plants when they're actively in a growing phase, when they're not too woody and like seeding out. How long it's going to take is going to depend on how much cellulose and lignin that you've got and on your climate. We're finding generally like in the New Zealand temperate environment, that might take 6 months. Up in Canada, it might take a year. So just take a look and see. But yeah, less than a year. But yeah, like I said, the research said 25 kilos a hectare, like if you were just blanket spraying. But you're not going to do that. You're going to be spot spraying. So, you know, a little can go a really long way.
And I think you've answered that question about cakeweed.
Yeah. Because
that's a nitrate accumulator. So
yeah,
the tea can cut. Does it work on kikuyu?
I don't know. We haven't tried it on kikuyu. I guess as long as you're harvesting just that, and you know, kike we know is a calcium indicator, and you can tell by that root system as well if you take a look at those roots. It doesn't take up any iodine, is that right? I'm trying to recall. I haven't dealt with kikuyu for so long. Yeah, it's got some weird root system. Yeah, so not all plants are dynamic accumulators, and that's where you're asking the question of what is it specifically that's happening in here. Is it a biological indicator? And you'll need to do the testing yourself. But, you know, a lot of these grasses, when they really get a hold, and, you know, it's fairly primitive, so potentially it's on your bacterial index instead. So instead of a weed extract, we'd probably be looking at addressing the biological community. Actually, I don't know if that's true. Trying to think of the places where we've had a lot of kikuyu.
It's a tricky one.
It is a tricky one. Good one to look into, though, Wilhelm, and just, you know, do the testing, see what you can find, and then let us know.
Well, I think we're at the end of the questions, amazingly. So I want to say thank you once again, Nicole, for an absolutely brilliant presentation and Q&A. For our audience's benefit, Nicole has very kindly agreed to do an audio version of the webinar in a podcast, which we'll be recording next week and we'll release at the end of August. So if anyone's like me, I need to hear things 2 or 3 times for them to sink in. And sometimes sitting down to watch a webinar is not always possible, but a podcast is, you know, really good. You can listen to it on the tractor or in the car. So hopefully we can reinforce the information via the podcast soon. So looking forward to chatting again for that. But I'll let you go now, Nicole. It's late there. I don't know if you got to finish.
20 past 8.
Oh, and hopefully you got to finish your dinner before. But
yeah, no,
we'll see.
So lovely to talk to you.
And
great questions, everybody.
And also, just for a reminder for our audience, actually, and it's interesting that Nicole mentioned Johnson Su compost in her presentation. We are very delighted to be able to announce that we will be hosting a webinar with Dr. David Johnson and his wife, Wei-Chin Sue Johnson, coming up on August 16th, I believe. So I'll be advertising that shortly, and I think that'll be really, really interesting to see what he has to say. For everyone else, I think that's it for the night. I might just get you, if you wouldn't mind, to complete our evaluation. As you know, I don't like to let you get away without filling in a questionnaire, but it's really important for us so we can know that we're still providing the content that you want. And look forward, I'm going to say goodbye now and look forward to seeing you all next time.
Thanks, Kate. Thanks, everyone.
Thanks, Nicole. Bye.
Rocking. Bye.
Bye.